In the third section of Chapter 2, we'll talk about enzymes as biological catalysts. Here we'll talk about the effects of the enzymes on the kinetics of chemical reactions, but not change the equilibrium between the reactants and products. We'll talk about the mechanisms of how these enzymes work, and we'll talk about the difference between an enzyme versus a coenzyme, and we'll talk about why the regulation of the activity of an enzyme is very important to the cellular function. So as we talk about enzymes, they are going to have two fundamental properties. One, they increase the rate of chemical reactions without themselves being consumed or permanently altered by the reaction. Otherwise, they would not be very efficient. And second, they increase reaction rates equally from the decomposition as well as the synthesis without altering chemical equilibrium between the reactants and products. Now looking at this chart here, you can see how the activation energy needed, or free energy needed to catalyze an uncatalyzed reaction is significantly greater than one that's going to be catalyzed by an enzyme. As you can see by the red line for the uncatalyzed reaction, more ATP energy is used versus the catalyzed reaction shown by the purple dotted line. You can see that the energy activation is a lot less, so therefore more efficient. Now, the thing to look for is your starting free energy level at the substrate to the product is going to be independent of whether the reaction is catalyzed or uncatalyzed. So again, the net free energy change from substrate to product, in this case, will be exergonic. So free energy will be released, will remain the same. So again, it doesn't matter whether the reaction is going to be catalyzed or not. The net energy at the end of this particular reaction is going to remain the same. Now, enzymes tend to be very specific for a certain type of reaction. They mostly handle just one reaction, but may, of course, handle two, oftentimes maybe the reversal of that action. Very simply, it offers a surface area for the substrates to bind both to each other and also the active site of the enzyme, which will form a conformational change to create your product that you see at the end of the slide here. Now, it was thought before that this was a lock and key type of fit, where very simply a substrate will simply just bind to the enzyme with the enzyme not changing very much at all. This goes back about 20 years back when I went to school, but now we have more understanding of exactly what conformational change we have both at the substrate and also the enzyme. So this was known as an induced fit. So the substrate will act upon the enzyme as much as the enzyme is going to affect the substrate and turn it into products. So both have a conformational change. Now a few slides back we talked about the different types of amino acids found within the polypeptide chain. Some are nonpolar, some are polar, some will be acidic, some will be basic. Each one of these will have different interactions depending on the particular enzyme. In this case, we look at chymotrypsin, which undergoing a hydrophobic interaction here, will bind one of the nonpolar amino acids just adjacent to where the peptide bond needs to be cleaved. So you can see the direct interaction between the enzyme itself, the specificity of that particular enzyme to a particular portion of the particular polypeptide chain to be cleaved. While looking at trypsin on the right-hand side here, you see the binding pocket has a negative charged amino acid known as aspartate, which is going to have an ionic interaction with that particular base that we're going to find within its polypeptide chain, again, just adjacent to where they want to cleave that particular peptide bond. So again, the specificity of each individual enzyme is very important to cleave a particular portion of the polypeptide chain. Now we'll examine this particular active site on chymotrypsin a little more closely. Dealing with the binding pocket and the different amino acids found within the enzyme itself that help us cleave this particular polypeptide chain into its smaller subunits. So this is a smaller picture. We're going to look at each step through here as we look at different transitional stages for both the polypeptide chain and the enzyme and see what steps we take in order to cleave this particular peptide bond. See in the first step here that serine is going to donate its hydrogen to the adjacent histidine in preparation to have the hydrogen actually bound to the cleavage site. But to help with this, the aspartate is going to help stabilize the histidine with the extra hydrogen through an ionic reaction. Next, you see the hydrogen. The extra hydrogen with the histidine will be donated to the polypeptide chain, cleaving this particular area from the C-terminus to the cleavage site, which now will produce a new N-terminus as seen in the new amino acid group on the left-hand side of the free polypeptide. Notice here, as the free peptide is released, notice how the aspartate is going to help further stabilize histidine, who is still looking back for its donation of another hydrogen to stabilize this portion of the enzyme. In the second part, you see a water molecule that will start to bind or become attracted to the histidine found here on this side of the enzyme. The water molecule binds with a hydrogen bond to the histidine, and then it will catalyze to donate its hydrogen to the histidine, while the hydroxyl group will go to the rest of the amino acid chain just off the serine portion of the enzyme. So in reverse action from one of the first steps we saw of this particular reaction, the histidine will now donate its hydrogen back to serine, and then action there will help release the rest of the protein from the active site. Next, we talk about a particular subset of molecules called coenzymes. These are basically prosthetic groups that are bound to proteins to actually enhance the critical reaction that's done in the catalytic process. Most of the time, these could be small metal ions, such as iron that we see in hemoglobin and myoglobin, or in other species, zinc or iron, that are bound to enzymes and undergo another conformational change in a reversible manner. So these are recycled back and forth. One of the ones that you're probably most familiar with is one we see as an electron shuttle. This is nicotinamide adenine dinucleotide, or NADH, as it goes back and forth from the oxidized to reduced states, reduced as NADH, and then oxidized as NAD+. While NADH is very integral in different cell cycles of cellular respiration, including glycolysis, the citric acid cycle, as well as the electron transport chain, it's a major component to shuttling or constantly doing the oxidation reduction reactions all throughout those particular cycles. The other ones you may be familiar with would be the FADH that you also see in cellular respiration, and also the NADPH that you see often in the light cycles found in photosynthesis. See the example of a net effect of the oxidation reduction reactions using NAD plus and NADH. So we have two very simplistic substrates here, S1 and S2. S1 starts as reduced, so it has an electron, it will donate it to the NAD plus. There will become oxidized and therefore left with NADH plus a hydrogen. That same NADH plus a hydrogen will now take an oxidized S2 and reduce that, and become oxidized itself to NAD plus. So the net reaction is reversal here of a started reduced S1 and oxidized S2, whereas we'll complete it now with a reversal into the S1 being oxidized and S2 now being reduced, all because of the NADH, NAD plus electron shuttle. Now, aside from transferring electrons, there are other coenzymes that will actually transfer certain types of chemical groups from one substance to the next. Some of these related vitamins should be very familiar to you from the size of the cereal boxes, and these are intimately related to different types of coenzymes. So NAD plus and NADP plus are closely related to niacin, of course, the ox-redox reactions. FAD, also used with riboflavin, or otherwise known as vitamin B2, also with redox reactions. Thiamine pyrophosphate is very closely related to thiamine, or vitamin B1, useful for aldehyde groups. Coenzyme A, pentaphenate, for acyl group transfers. Tetrahydrofolate, which is going to be related to the vitamin known as folate, for methyl group transfers. And pyridoxal phosphate B6, pyridoxal, is used for transamination, so moving one amino group from one section to another. And of course, biotin for carboxylation of the carboxyl group on the opposite end of your amino acid chain. And most of these enzymatic reactions cannot run amok on their own, otherwise we have too much of a shift from equilibrium of too many products or too many substrates, depending on which direction the enzymatic reaction is occurring to. So normally, if there is enough binding sites and there's enough substrates, the reaction will continue to occur. However, if we need to somehow shut this down or slow down the enzymatic reaction, oftentimes you'll have a competitor will basically inhibit that particular active site, therefore making it unavailable for any particular substrates to bind to it. This is known as competitive inhibition because you're blocking the actual active site for the actual substrates to bind to. The other way we shut down enzymatic reactions is to have what's known as allosteric regulation, where an inhibitor will actually bind to an allosteric site, which is going to be a non-active site, which therefore causes a conformational change, which closes off the active site. So both of these will help inhibit or slow down any type of enzymatic reaction. So either by direct inhibition or by indirect inhibition, also known as allosteric inhibition, but both are successful in closing off active sites to any particular substrates. Now, aside from having too much of a shift in equilibrium from either the formation or the catabolization of certain types of products. The other thing the cell is trying to do is also regulate how much ATP is being used and not being wasteful in certain types of reactions. So it often will employ what's known as feedback inhibition. This is very similar to the feedback loops we saw with the homeostatic processes. In this case, we see threonine, which is going to be catabolized by threonine deaminase into alpha-ketobutyrate, which the end product is going to be isoleucine. Now, isoleucine is going to act as an allosteric inhibitor. So once you have an abundant amount of isoleucine, it'll actually deactivate allosterically threonine deaminase. So therefore, you will not continue to break down threonine until the isoleucine levels start to drop again, whereas then the isoleucine will be released from threonine deaminase to start producing it again. So it's like a feedback inhibition loop. So when you have too much of it, it'll start to slow things down. It'll start to inhibit the threonine deaminase. And then once there isn't enough levels of isoleucine, gets too low, then it'll reactivate and also convert a threonine back into isoleucine. So in the last slide of this particular section of the chapter, we're going to talk about activation of different types of enzymes, whereas before we talked about different types of inhibition or feedback inhibition to basically slow enzymatic reactions down. Basically what we're looking at here is looking at serine under letter A, which is going to become phosphorylated by the addition of a phosphate group donated from ATP. What this will do is increase a negative charge on the serine, which if it's any part of any particular type of an enzyme or a protein, it'll cause a conformational change. In this case, it will try and open up certain active sites. Now more specifically, if we look at, say for example, the areas of a muscle, which will have its own glycogen store, when it comes in contact with epinephrine, epinephrine will activate the phosphorylase kinase protein, which will basically cause a conformational change in this particular enzyme to open up its active sites to bind with glycogen, cleave it, and allow glucose 1-phosphates to be released to be used the rest of the muscle cell. Another type of interaction you see with this type of phosphorylation is with any kind of conformational change. We're opening up active sites in the muscles with troponin, tropomyosin, and addition of calcium and ATP, which can further generate the actin myosin filaments to move when doing any kind of contracture of any particular muscle group, which we'll get into in a few further chapters later on in the semester. This will finish the third section for the enzymes, and look to do the fourth one with the plasma membrane. It'll be relatively short, so you should be ready for the diagnostic quiz after the next section.